Welcome, intrepid students, to the fourth installment in your first week of the life history of mammals. So today is a short lecture, I promise, where we'll focus on important mammalian adaptive radiations during the evolution of mammals. So I'm going to be summarizing Chapter 3, which is entitled Phylogeny and Diversification of Mammals, specifically timing of mammalian radiations. So we're definitely not going to start here. So this picture illustrates the Pleistocene megafauna, a radiation that included some really impressive species like the giant ground sloth here, Megatherium, the massive Glyptodont, which was an armadillo, and then of course the impressive saber-toothed cats, the Smilodon. So you can see these wonderful Smilodon skulls, as well as dire wolves and a lot of this Pleistocene megafauna at the LA La Brea tar pits in Los Angeles. So these enormous species, they only went extinct about 11,000 years ago, which is really a geologic blink of the eye. So our colonization of the globe, ours meaning Homo sapiens, likely contributed to many of these species' demise. But I'm going to return to the Pleistocene at the end of this lecture. So as you'll surely recall from our last lecture, we started way back here in the Carboniferous period with those very first amniotes. And then I spent most of the lecture in the Permian and the Triassic. So today we are going to start from the late Triassic here with the divergence of the monotremes and the therians. We're going to talk a bit about the end Cretaceous, as well as the Eocene, and then we'll finish up with the Pleistocene. So please note those last two time frames, the Eocene and the Pleistocene, those are epochs. And an epoch is even shorter than a period, and of course considerably shorter than an era or an eon. Evolutionary biologists call the clade, including the most recent common ancestor from all of the living lineages, as the crown group for that taxon. Thus, the crown group for the class Mammalia represents that common ancestor of the living lineages, which are the monotremes here, the duck-billed platypus, the spiny echidna, as well as the therians here. So the metatheria, those are the marsupials, and the eutherians are the placentals. So you can see when those three living lineages, extant lineages, converge, that represents the most recent common ancestor, and these are deemed the early crown mammals. So this lineage diverged before the evolution of the crown group. So we're going to refer to this lineage, the Mammalia formes, as the stem group. Okay, so the recognition of a crown group, at least relative to stem group, is really essential for those molecular analyses that are estimating divergence times. The oldest monotreme yet recovered from the fossil record is Steropodon here. That fragment right there, dating at 110 million years ago and recovered from Australia. The oldest metatherian, and again, the metatherian includes the present-day marsupials as well as the non-marsupial metatherians like the amazing Sparassodonts of South America, and we'll get into that adaptive radiation soon. The oldest metatherian is here and is named Cenozoic. It dates at 125 million years ago and was actually recovered in China. And then lastly, we have Jeremiah here on the bottom center. This is the oldest eutherian, and it's actually coming in at 160 million years ago. So we know, given that the common ancestor of these lineages must be at least as old as the oldest fossil, Jeremiah, here, we know that the crown group of mammals are already in existence a minimum of 160 million years ago during the Jurassic. However, the relative scarcity of mammal fossils throughout most of the Mesozoic means that monotremes, metatherians, and eutherians, they didn't occur during the age of dinosaurs at anything like their current diversity. So, when did the great adaptive radiations of mammals take place? There are two primary sources of evidence: the fossil record as well as molecular clocks, and they often have produced very different answers. Over the past 40 years, evolutionary biologists have been investigating the possibility that some evolutionary change, mutations, may occur in a clock-like fashion, in that these mutations are going to occur at a regular rate. Over the course of millions of years, mutations may build up in a given stretch of DNA at a reliable rate. So, for example, the gene that codes for the protein alpha-globin, which is a component of hemoglobin in animal red blood cells, it experiences a base change rate of 0.56 changes per base pair per billion years. So if this rate is reliable, then this gene could be used as a molecular clock, looking at the different number of mutations in this protein across species. So when a stretch of DNA does indeed behave like a molecular clock, it becomes a powerful tool for estimating the date of lineage splitting events, like you see here. So imagine we've got two stretches of DNA, two different species represented by the red lines that share a common ancestor here. So we've got four bases that are different between these two species. You can see this is a C, whereas this is a T. Okay, this is a G, where this is a T. Okay, we're going to hypothesize that the entire length of DNA is going to change at a rate of approximately one base pair per 25 million years. So, 4 base pairs, 25, 50, 75, that's 100 million years of evolutionary distance. That said, you've got two lineages. So this represents 50 million years, and this represents 50 million years, for a total evolutionary time of 100 million years. We can then infer that the common ancestor lived 50 million years ago. So that is an accurate but very simplified version of the methods used to generate the phylogenetic tree that you see before you. So this was published by Meredith and colleagues in 2011. The figure is in your book. Here, researchers undertook a super matrix analysis of 164 mammal species, representing 97 to 99% of extant mammal families, and DNA sequences from 26 protein coding loci. That is 35,603 bases. So a very impressive data set. They use maximum likelihood and Bayesian analyses to produce the most parsimonious phylogenetic tree, making the least assumptions, and that's the tree that you see before you. So Meredith et al. 2011, they estimate that the monotremes diverged from the theria somewhere between 203 and 238 million years ago. There's considerably more precision in their estimates of the divergence between the metatheria and the eutheria, and they estimate that between 168 and 178 million years ago. So that's very consistent with Jeremiah, the oldest eutherian fossil, which dates at 160 million years ago. Very important to take a moment here and stress that phylogenetic divergence, or speciation, is driven by and maintained by reproductive isolation. And this reproductive isolation was facilitated by the breakup of Pangaea. Remember, Pangaea is the supercontinent that existed right around this time at 200 million years ago. Recall that at 200 million years ago, the Therians likely hadn't evolved, and if they had, they certainly weren't widely distributed. But the Prototheria, what are to become the monotremes and the metatheria, are likely already in Australia. By 180 million years ago, the primitive eutheria are in West Gondwana, and the monotremes are in East Gondwana, and then there's metatheria in both. Okay. By 65 million years ago, the eutheria have radiated in present-day Africa 65 million years ago, so they're about to go gangbusters. Notice that Madagascar has already split from the African continent. And the metatheria are in Australia, a green Antarctica, as well as South America. Wait, metatherian marsupials in South America? You may have thought that marsupials only lived in Australia. Well, we're going to talk more about this next week. But for now, please put me on pause, and I want you to check out this amazing 10-minute video. It's entitled, How South America Made the Marsupials. The YouTube video is embedded in Canvas, so take a moment and check it out now. A really consequential evolutionary event occurs 125 million years ago. And that is the evolution of the Angiosperms, the flowering plants. So this period here, denoted by light green, is referred to as the Cretaceous terrestrial revolution. But following the evolution of flowering plants, you have the coevolution of all of those arthropod pollinator species. You see this incredible explosion of beetles, bees, butterflies, moths, straight up flies, and this is occurring from the middle Cretaceous all the way to the late Cretaceous, when the dinosaurs still ruled. As you would likely predict, if you have this incredible explosion, this incredible diversification of arthropods, of insects, that suddenly represents lots of new ecological niches for insectivores. And we see at this time this simultaneous diversification, radiation of both the metatherian and the eutherian insectivores, as well as these lineages We see diversification in the multituberculates and the dryolastoids. However, these lineages are bound for eventual extinction. My larger point here is that the Metatherian and Eutherian lineages were already diversifying during the time of the dinosaurs before that hellish day 66 million years ago, which radically alters the evolutionary trajectory of life on planet Earth, and without which we wouldn't be here. One fateful day, 66 million years ago, the world was changed forever. An asteroid estimated to be 6 to 9 miles in diameter, the size of Manhattan Island, traveling at an estimated 44,712 miles per hour, smashes into our planet with an energy equivalent of 100 trillion tons of TNT. That's 21 billion Hiroshima atomic bombs. It generated a cloud of superheated ash and steam, killing everything for hundreds of miles, and triggering perhaps the most massive earthquakes the world has ever seen, tsunamis with 100-foot waves around the world. Further, trillions of tons of material are ejected into the atmosphere, initially raising global temperatures and causing raging wildfires around the globe. The water, air, and soil were poisoned with cyanide and heavy metals, nickel and lead that composed that asteroid. The sulfur released into the atmosphere caused acid rain, and then we have a global cooling known as a nuclear winter that probably lasted for decades. The loss of photosynthesis reverberated throughout the food web, and all of the large-bodied animals, greater than 55 pounds, with the exception of the crocodilians and the sea turtles, who managed to make it through somehow, all of the large-bodied animals over 55 pounds blink out. It was hell on earth. Carl Sagan famously said, "Extraordinary claims require extraordinary evidence." Well, here's the evidence. This is the impact site for that asteroid. It's known as the Chicxulub crater, and it's hidden beneath the Yucatan Peninsula and the Gulf of Mexico in present-day Mexico. If you've ever been to Cancun or Cozumel, it's on the Yucatan. So this buried crater is over 93 miles in diameter, and it's 12 miles deep, well into the continental crust. Further, the end Cretaceous is marked by this very thin, dark layer of iridium that's been detected at hundreds of sites around the world. Iridium is a very rare element in the Earth's crust, in the sedimentary rock, but it's abundant in meteorites. And this iridium is found directly above the Cretaceous fossils. So as this meteorite struck with this incredible velocity and impact, it vaporized, sending that iridium into the atmosphere and eventually raining down in this sedimentary layer. It's now found at 100 times the background rate of iridium in the other sedimentary layers. Small, nocturnal, insectivorous mammals somehow make it through the end Cretaceous extinction event. And then they are going to radiate into an incredible diversity of forms. So most mammalian orders are now freed to begin differentiating, radiating in the early Cenozoic, because as you may have likely heard, nature abhors a vacuum. So this mass extinction event, as devastating as it was for life on our planet, it allowed for the age of mammals and eventually Homo sapiens. Most of the extant orders of mammals are recognized in the fossil record by the Eocene epoch. That's 55 million years ago. And the vast majority of families, of course, families below the taxonomic rank of order, most families are recognized by the Miocene epoch, which is 23 million years ago. So for the next 6 weeks, we are going to systematically cover 21 of these orders, and we're going to relate those orders to significant attributes of mammalian life history. So it's going to be an exciting ride. I promised at the outset that I would return to the Pleistocene megafauna. So if you're interested in woolly mammoths and Smilodon, the saber-toothed cat, and giant ground sloths, giant armadillos, all these incredible animals that were roaming North America, I've embedded one of my Estrella Mountain Community College YouTube videos into your Canvas page. It is not required. You don't have to watch it, and you won't be assessed over it. But if you're interested, it's about a 20-minute summary on a new debate in conservation biology revolving around this technique referred to as Pleistocene rewilding. So given new advancements in biotech, is it appropriate to bring back highly influential, ecologically influential species that we likely drove into extinction? So more recently, the passenger pigeon and the thylacine, or even the woolly mammoth, which only went extinct about 4,000 years ago. So if you're interested, check it out. I've got a couple of references that are not in your book that I used some of their graphics. I've got Benton is a new one here, 2022, as well as Gross-Nickel et al. From 2019. I just thought I'd point those out. I hope you learned something, and I'll see you next time. Cheers.